Okay, so hello guys and welcome back to Excelsior Academy. So we are starting these classes with the concepts of small molecular biology, and the first topic what we are going to study is DNA replications. So in this also, what I'm going to do is whatever we are going to study, what kind of interview questions will be asked, okay, in future, those also we are going to discuss. So you don't have to prepare separately for your interviews, okay. So in this session, we are going to talk about the basic chemistry of DNA synthesis. Like in the DNA replication, क्या क्या हम पढ़ने वाले हैं? Basic chemistry, function of the enzymes, what all are involved in your DNA replications. Of course, the replication fork, which is very important. This is like every time they have asked question based on this replication fork. Then origin of replication. In this also, मैं अभी आपको बता दे रही हूँ. In the case of prokaryotes, we always ask what is the sequence, okay, which gives you the signal that these are the origin of replication, or what are the termination sequence. So those are the few sequences which you are going to by heart. आपको by heart करना पड़ेगा. Yes. Okay. So the replication in prokaryotes, and before this, basic things. It is carried out by the enzymes. We all know that those are the DNA polymerase. Okay, and in DNA polymerase, in the case of prokaryotes, there are at least 3 types, mainly DNA pol 1, 2, and 3, which we will be discussing in detail. So when I say in detail, it means we are going to study its structure also. What kind of cofactors or the prosthetic groups are used by those enzymes, how they are going to perform the function, the basic chemistry behind the protein and their targets or the substrates also we are going to study, okay. So they use DNTPs as the precursors, and the process starts with the separation. So now we know that this is because of the helicase enzyme. Okay. Now, can anyone tell me what is the name of the helicase? Sorry. Helicase is helicase only. And then after the DNA is getting replicated, they are going to be twisted, if you know. So this is how what happens when the helicase is opening the structures of the DNA. These portions are getting tightly twisted, right? ये क्लियर हो रहा है? जैसे जैसे आपका यहाँ पे रेप्लिकेशन फोर्क ओपन होगा, what happens to this portion? यहाँ पे टाइट होता रहता है. Are you able to, है ना? What about the others? Akshaya, Shruti?
Yes, ma'am. Yeah.
Yes. Yes. Now, so we have got certain enzymes called topoisomerases. Okay, don't worry, I'll take in detail 1 and 2. What I want to convey over here is that topoisomerase 1 and 2 in case of eukaryotes. In the case of bacteria या prokaryotes, we call them as gyrase. ठीक है? ये सारी चीजें हैं जो आपको याद रखनी है. So you can make notes also, or you can listen to the recordings later. Okay. Of course, we know that DNA replication is semi-conservative replication. I'm not going to take because these are very basics. We know about the ATGC base pairing. AT is how many hydrogen bonds are there, guys?
2.
Yes. So we have got in AT, we have got the 2, and in GC, we have 3, right? Now, based on this concept, okay, the Chargaff's rule, you should know that the number of purines divided by number of pyrimidines should be how much?
1. Close to 1, ma'am.
Sure. Yes, it is 1 because जितने A होंगे उतने T होंगे, जितने G होंगे उतने C होंगे. So in other forms like in the textbooks and all, you can see that A plus G divided by C plus T or T plus C, whatever, it is going to be 1. This is something you should always remember. Okay, and this one also you need to remember. Why I'm saying is that though it is very simple concept, there are certain numericals which needs these 2 concepts. Okay, for example, let me give you one example right now itself. So let's say that you have got 100 base pair DNA. Okay, Now, I am saying that in the 100 base pair DNA, you have got 20% adenine. I am asking you total number of hydrogen bonds. Now, can you solve it? Is the question clear to everyone?
Yes, ma'am.
Yeah. Okay. So what should be the first step? Tell me. I'll tell you whether you're going wrong or not. Anyone can tell me the answer. Whoever thinks is the right. It's okay. Don't worry. So if I have got 100 base pair and 20% adenine, so how much? 20% of 100 base pair is how much? Simple math.
20.
Yes. So what does it mean? So in 100 base pairs, I have got 20 adenine. Correct? From this, what can you conclude?
Thymine is also 20.
Yes.
Thymine 20.
Yes, very good. So what are the remaining GC? How much it will be?
30, 30.
Yes. So it's simple maths, right? 20 is adenine, 20 is thymine, total 40. Out of 100, remaining is 60. So half each is going to be your G as 30 and C as 30. Now tell me how I'm going to calculate the total number of hydrogen bonds over here.
I think 20 into 2, ma'am.
Adenine, thymine, how many are there? 20.
2 hydrogen bonds.
So you are saying, okay, Chandni is saying 20 into 2. So similarly, 30 into
3.
3. So how much should I get?
And 40 and 90. In
Yes.
case of adenine, 40 and 90.
So total?
130.
130. So how many of you think it is correct? Hmm? Yes. Everyone is okay with this? कहीं पे कुछ doubt आ रहा है?
Yes, ma'am.
Okay, what is the doubt, Chandni?
No, ma'am, I got clear that.
हाँ.
No,
Anyone
no doubt.
else? Simple लगा?
Yes, ma'am. Yes, ma'am.
Okay. Even though it was simple, but we should know when to use what. Okay. आगे जाके और भी complicated होगा. But for your practice, like already there, it is there in my YouTube playlist, like how to calculate. So many twisted questions are there. But I'll bring more for our practice. Okay. So if you get time, you can just do those numericals from the playlist. There's a playlist by numericals of biochemistry and Mol Bio. So those you can do. Okay. Okay. So DNA replication, basic mechanism, first we are going to study, and then we'll see how the difference in the bacteria and eukaryotes it is going to vary. Okay. So first thing is first, how the DNA is being synthesized. So we know those are the basic two requirements, and those are the DNTPs. Okay. Now DNTPs means deoxy nucleotide. So we have got how many? 4. G, C, A, and T. इसमें भी you can't say that it is guanine. Okay, guanidine, cytosine, adenine, okay, adenylate, and thyminate. इस तरह से आपको बोलना है. Then they have 3 phosphoryl groups that are attached via the 5' hydroxy group and 2' deoxyribose. क्यों? Because 3' में तो OH है. Getting the point? We always talk that it is 5' to 3' direction. 3' में तो आपके पास क्या है? You have got the OH group to which the phosphate group attaches. Okay. Now, one more thing. This is your nucleotide. Nucleotide means having base, sugar, and the 3 phosphates. Correct? So this is your NTP, nucleotrophosphates. Now, the first phosphate which is attached to the 5' OH, okay, is called alpha, then beta, then gamma. Is this much clear?
Yes,
Yes,
ma'am.
Yes, ma'am.
One more thing, the bond which you're going to break, okay, the bond which you're going to break, OH and phosphate के बीच में जो bond बना है, those are called phosphodiester bond. Okay, so the bond which you're going to break over here or here or here, let us say that this is first, this is second, and this is third. Now tell me which breaking of the bond is going to give you the maximum energy. अगर मैं break कर रही हूँ at 1, 2 and 3, okay, which one is going to give me the maximum energy?
Alpha, ma'am.
Okay, she says alpha. What about others? It's okay, galat hone do. But I just need to know how you guys think.
Beta.
Okay. And I can't see who कौन क्या बोल रहा है. तो एक ने बोला alpha, एक ने बोला beta. Okay, don't say alpha beta. It's either 1 or 2. Okay, yeah. Okay, so I'll give you the answer now. The answer is that you're going to get the maximum energy by breaking second. Now I'll give you the reason at the end of this lecture. आपको समझ में आएगा. And here what we are going to learn is something called thermodynamics, जो आपने स्कूल में पढ़ा होगा. Okay, so this answer you will get at the end. Why? Okay, नहीं clear होगा, so stop me and let me know so that I can clear again. Okay, so second essential substrate for the DNA synthesis, single stranded DNA, so that आपका primer and template junction बने. So पहला क्या हो गया DNTP. Next we have got primer and template junctions. Okay, but again primer and template junction, it has got the key components. What are those? That it should have a primer having free 3-OH. Sorry. Now, what is the chemistry behind this DNA synthesis? Can anyone tell me? आपको क्या लगता है? What is chemistry behind this? This is one of the interview question. Or in another words, what is the basic mechanism behind DNA synthesis? Yes, Tanvi, can you give the answer?
Ma'am, I think it's like protein synthesis mechanism.
No. So, okay, if you say protein synthesis, what is happening during protein synthesis, Chandni? What is the basic mechanism behind protein synthesis?
tRNA.
That is like part
tRNA
of...
attaches the.
No,
Yeah.
Chandni. That is something translation happening. I am asking the basic mechanism, the chemistry behind protein synthesis. When a bond is formed, okay, tRNA is bringing the amino acid. So amino acid bond is forming, right? Which is called peptide bond. What is the chemistry behind that peptide bond formation? See, this is why the exams become difficult to clear, even though questions is very simple. Okay, I'll give you the answer. Don't worry, guys. Don't panic. Okay. So the answer is very simple. So for protein or peptide bond formation, if you know the structure, there is always CONH bond formation, right? This is only happening if you have got the acid and amino group. So what is happening over here? I should make like this. OH and one H is taken. So what is happening over here? Removal of water molecule. And this process is called dash. What is that process called? Removal of water molecule is called
condensation.
Dash. Very good. It is called condensation. Okay. Now in future, if anyone is asking you what is the basic mechanism or the chemistry behind protein synthesis, don't say protein, just say peptide synthesis, then it is condensation reaction. Similarly, keeping this concept in the mind, now tell me what is the basic mechanism or the basic chemistry behind DNA synthesis? Yes. अब इस क्लियर हो गया ना मैं क्या पूछना चाहती हूँ?
Same condensation.
but before that यहाँ पे क्या है, directly you have got COH and NH brought together with the help of the tRNA, right? यहाँ पे कैसे होगा? You need to bring DNTP from somewhere far, so it should have something over here to attract. चलो panic नहीं करेंगे, let's go ahead. There's something called in chemistry, it is called SN2 mechanism. सुना हुआ लग रहा है? Yes. Anyone who can explain SN2 mechanism क्या है? So when you talk about SN2 mechanism, आज तुम्हारा यही homework होना चाहिए. That what is SN2 mechanism? You need to know what you mean by electrophile. What do you mean by nucleophile? Okay. So if you do understand what are these, then it becomes easy to understand SN2 mechanism. Anyway, so we'll have this discussion today and you can do yourself also for better understanding. Okay. So the phosphodiester bond is formed by an SN2 reaction, jahan pe hydroxyl group at the 3' end of the primer is attacking at the alpha phosphoryl group of the incoming nucleotide. Clear? So if we go back, you can see that here you have got the OH group at the 3' end. This O is going to act as the electrophile. Okay, oxygen has got the negative charge, or that is electron loving it has got, right? So it is going to attack at the DNTP. And where it is going to attack? Where it is going to attack? It is going to attack at the alpha. Now where is the alpha? This one is your alpha, this one is your beta, this is your gamma. This phosphate is your gamma, right? So where it is going to attack? Which bond? As I mentioned before, this is 1, this is 2, this is 3. So at which number it is going to attack? The 3-OH of the primer. As I said, it is going to attack at the alpha of the phosphate of the incoming nucleotide. Yes. Yes, Shruti, what do you think? कहाँ attack करेगा? 1, 2 or 3?
1.
No, dear. It is going to attack at 2 because see, that's how 2 phosphate will be removed and 1 phosphate will be there with the ribose and the base, so that this particular nucleotide get incorporated in your DNA strand. अगर वो 1 पे अटैक करेगा तो क्या होगा? Only ribose sugar and base is there, but we know that in the DNA you have got phosphate also in the backbone, right? So it is going to attack at the second, so that your double phosphate, जिसको हम PPI बोलते हैं, this is also known as pyrophosphate, this will be released. Now is this much clear to everyone? Yeah, okay. So that was your SN2 mechanism. So what is happening? See, OHO, it is attacking at the alpha phosphate. So the bond is broken and PPI, the beta gamma phosphate is removed. Okay, and that is your happening over here. Now this is also, of course, with the help of the water molecule and all, we'll study all those, like how in the catalytic pocket of the DNA polymerase. So DNA polymerase is replicating your DNA, right? So how DNA polymerase is how it is making them possible. Now PP, that is your beta and gamma phosphate, does not exist in the cell like this as pyrophosphate. What is happening to the PP? Pyrophosphatase, the enzyme, is breaking down into 2-phosphate molecule so that it is ready for the phosphorylation. So you know that in the cell, in the cytoplasm, in the nucleoplasm, you have got the kinases. What is the function of kinases? They are the enzymes which always adds phosphate group, okay, to your substrate. Jaise, for example, you have got hexokinase, glucokinase, glucose 6-phosphate in the glycolysis. Okay, so they are the kinases, and these kinases add phosphate group. ये phosphate कहाँ से मिल रहा है? Because of this. Okay, is this much is clear how one nucleotide has been added into the DNA? At any step, any doubt is there? क्लियर? अगर कहीं पे
Yes,
रिपीट
ma'am.
करना है, जस्ट लेट मी नो, आई कैन रिपीट, नो प्रॉब्लम, बट दीज बेसिक अंडरस्टैंडिंग इज इम्पोर्टेंट. Okay, now this is also one of the interview questions. Why DNA synthesis is irreversible? Now what does it mean? अगर आपने कोई bond बनाया है, so there are chances that it can be broken down, right? So why it is irreversible? So that answer is over here. Now this is what the answer for your that question, like the PP bond which I was talking about. Okay, why it has got the maximum energy. Okay. So मैंने question क्या पूछा था आपसे? Which bond has got the maximum energy? Either 1, 2 or 3. Right? This is alpha between the and the phi dash OH over here. This is between the beta and gamma over here, right? So ये bond कौन सा रहेगा? So what happens is that when there is an attack, what happens? This much is gone. Your nucleotide has been used. Now we have got this two. That is the beta phosphate and the gamma phosphate. Now what happens is that when this gamma and beta, the phosphate, this bond is broken down, tremendous amount of energy is released. Okay, now that energy is so high, which helps in the DNA synthesis, that is, it makes the delta G very, very negative. And you should know that in the thermodynamics, when the delta G, which is also known as Gibbs free energy change, right? When it is negative, the reaction is going to be spontaneous. Okay. Now, for DNA synthesis to be irreversible, again, the delta G should be very high. Like suppose this is -7, it should be more. Okay. Then only it will be broken down easily. But it is not possible कि आप इतनी एनर्जी कहाँ से लाओगे, right? That's why it is irreversible. Yes, am I making sense to you guys? Okay, I'll explain this concept again. Okay, so you know that you have got the growing chain of your DNA. So this is your nucleotide. This is N number has been added. So when you have incoming nucleotide, another N+1, and what you get is pyrophosphate. Okay, so once the nucleotide has been added to the chain, the delta G is your -3 kilocalorie per mole. Okay, which is less. But when pyrophosphatase breaks down this bond between the beta and the gamma, right? When it is breaking this bond, energy is released. So whenever a bond is broken, energy is released. When a bond is made, okay, energy is absorbed. So what happens? This 2pi. So during this pyrophosphatase, of course, breaking the bond means it needs water, which is called hydrolysis. This results in the good amount of energy. Okay. So overall, what is happening? You don't get a PPI. Okay. What you get is 2pi. PPI means there is a gamma beta together. 2pi means there is only 2 phosphate group present. Okay. Now, this breaking the energy, the breaking the bond between the beta and gamma phosphate is having high delta G, negative delta G. Okay. So what is happening? Which is correspond to equilibrium constant of 10⁵, which is high. Okay. So this will not result in the DNA synthesis ka irreversible. If you know the relationship between the delta G and the equilibrium constant, negative nRT, we make it, right? We write it like this. Then from this, you should understand that it is high to make the bond. Okay. So it is not possible for the enzymes to break immediately. So we have got different enzymes to do this reaction. Okay. So you have DNA, and that's why DNA is very stable. You can just take it out from the plant leaf and keep it at the room temperature. It is going to be stable. वो बिल्कुल खराब नहीं होगा unless until you put the DNases, the enzymes to break down the DNA, and that's why it is very stable, irreversible. Now it is clear to everyone. Yes.
Ma'am,
Yes, Akshaya.
ma'am, why not the bond is broken between the first position or the third position, only in the second position?
Array, how it is
I did
possible,
not get it.
Akshaya? See, this is your ribose, okay? This is your base, okay? Now base means it can be AT, GC, whatever. This is your 2-deoxyribose, right? Here you have got your phosphodiester bond CH2, sorry, CH2O, यहाँ पे आपका phosphate हो गया. This is your alpha. Then another bond. This is your beta phosphate. This is your gamma phosphate. Okay. Now the question is, why this bond is giving the maximum energy? Sorry, this bond is giving the maximum energy. Why not this one, right?
Yes, ma'am. The first one and the third, the other.
1, this is 2, and this is 3. So this second bond is broken when there is a SN2 mechanism. Correct? That is your DNA synthesis. है ना? ये बॉन्ड ब्रेक होगा तभी तो ये न्यूक्लियोटाइड आपका डीएनए में जाएगा, राइबोस बेस एंड 1 फॉस्फेट. करेक्ट?
Yes, ma'am.
We don't break at this first position. There is no point breaking this. Otherwise, what will happen? The DNA is without phosphate, which is wrong DNA. It will go under mutation. That will be considered as mutation, and it will lead to cancer or apoptosis. That is different thing to study. So when this bond is broken down, this becomes your DNMP, that is monophosphate. So this goes into your DNA and gets incorporated into DNA synthesis. Okay. Now the question is, why breaking this bond releases more energy? Correct? This is the question, right?
Yes, ma'am.
See, it's because when this phosphoanhydride bond is broken. Now see, over here also, the water molecule was used. Okay, whenever you break any bond, water molecule is used. And to do this thing, that is phosphate and phosphate, the phosphatase in the phosphatase enzyme is going to get twisted a lot. Okay, for this to reach into the transition state, if you know that when there is a substrate, enzyme binds, it needs to reach its transition state, and then it becomes product and becomes a stable molecule, right? Now this transition state means having the maximum delta G energy, Gibbs free energy. Now this is brought easily with the help of the enzyme. Okay, so this is when the bond is broken with the help of the enzyme, the energy which is released, it makes it easy for the enzyme to convert them into two products. That is breaking the bond. In short, जब आप कोई चीज तोड़ रहे हो, आप उस सिस्टम का एंट्रोपी बढ़ा रहे हो. ठीक है? Whenever you break something, you are just increasing the entropy of the system. And that is why it is releasing the energy. In simple words, maths, how it is very simple. This is your thermodynamics equation. Delta G, delta H minus T delta S. Now, when you increase the entropy, what is happening to the delta G? When you increase this quantity, see temperature तो is going to be remaining constant, right? तो हमेशा वो 25 डिग्री सेल्सियस होगा या 28 डिग्री सेल्सियस होगा. This is not going to change, but delta S is changing. It means what? The number of molecules in the system you're increasing by breaking the bond, right? So delta S is increasing. Now, what will happen when the delta S is increasing? This quantity is going to be higher. This is delta H. The energy change is always fixed for the system, or it is less. तो क्या होगा? Total quantity is going to be negative delta G.
Yes, ma'am.
Right? So what is happening? So delta G is negative, and I know that delta G having negative means the system is allowing the variable. So it is going to be spontaneous. ठीक है? Now, दूसरा scenario समझाती हूँ. Same equation. delta G, delta H minus D delta S. You're making a bond. So that time your delta H increases and delta S decreases. Okay, आप कोई चीज जोड़ रहे हो. Entropy is degree of randomness. Okay, so when you're making a bond or you're making a protein, whatever it is, your delta S is decreasing. ये decrease हो रहा है, आपका delta H increase हो रहा है. So what is happening? Delta H means what? Free energy enthalpy change, right? Not free energy, sorry, energy enthalpy change. यानी कि मैं जब कोई बॉन्ड बना रही हूँ, इसका मतलब आई नीड एनर्जी टू ब्रिंग द टू सबस्ट्रेट टुगेदर. ठीक है, ढूंढ ढूंढ के उनको ला रही हूँ, उनको सामने ला रही हूँ. And when you have got two molecules together, you're bringing, it needs a lot of amount of energy. Okay, so delta H is increasing. Since you're decreasing the randomness, delta S is decreasing. Overall, delta G क्या हो रहा है आपका? Positive, right?
Yes, ma'am.
In the system, this is not spontaneous, but the reaction is taking place. वो कौन कर रहा है? Enzymes. Okay, अभी क्लियर है? थोड़ा बहुत?
Okay.
हाँ, सिंपल. इसी चीज को मैं एक और तरीके से बताती हूँ. So your DNA is very stable at room temperature. ठीक है? You can keep it at room temperature for 1 year, 10 years, 5 years, 25 years. Nothing will happen. Stable. But the moment you have got the enzyme called DNases, within a fraction of a second, your DNA is broken down into pieces. Why? Because enzymes are capable of bringing down that energy. Okay? And increase the delta S. And that's why now your DNA is not stable. And there are many factors which makes DNA very stable. Okay, so DNA is, if you have got, when we are going to study about the PCR, you will understand that how the Taq polymerase, the DNA polymerase which we use, it needs co-metals, sorry, metal ions, co-factors, etc., etc. We'll study in detail, don't worry. थोड़ा सा clear हुआ?
Yes, ma'am.
Yes, Akshaya? Okay, so good. It was a good question. So whenever you have such doubts, थोड़ा सा आपको basic मालूम होना चाहिए, because every time we'll be talking about thermodynamics. If possible, please study about the thermodynamics. I'm not asking you to remember the laws of thermodynamics, just the equations of delta G. So we have got different delta G equations. एक हो गया delta H वाला हो गया, एक हो गया जहाँ पे आपका minus NFE cell का है. Okay, this is also required. Another one is where there is a relationship with the equilibrium constant. Okay, so there are around 4 to 5 equations. Please study them, because every delta G change we will be needing in cell bio, mol bio, and all the methods and techniques what we are going to study. Everything is needed because for the numericals. Okay. Yeah. Anything else you guys need to ask? I'll be stopping here. Time was zyada ho gaya. Yes. Anything else? Any other doubts?
No, ma'am.
Okay. So I'll be stopping here now.